Today we're going to begin our lessons on genetics. We're going to have 3 parts of our genetics lesson. We're going to begin with talking about the basics of genetics with this lesson. Then we'll do some more advanced genetics. And finally, we will talk about genetic disorders. So let's begin our lesson today. Let's begin today by talking about the basics of genetics. So, what is genetics? In order for us to talk about genetics, what we need to understand is we need to understand what heredity is. Heredity is the way organisms pass their traits from parents to offspring. Scientists will track these characteristics or these traits in various populations. When we know a little bit about our genetics, which is the study of heredity, we're able to predict what some of these traits like eye color, hair color, intelligence, gender, and that will be. The study of these hereditary patterns is known as genetics. The father of modern genetics is Gregor Mendel. He was an Austrian monk, and he is really credited for beginning to investigate genetics. And he used pea plants in order to figure out the start of heredity and the study of genetics. And through studying these pea plants and doing his experiments, he was able to come up with a lot of data, and what he started to see connections from one generation of pea plant to the next generation of pea plant. Now, he controlled what pollination occurred. When we talk about cross-pollination is going from one plant to another plant. Self-pollination is where the plant pollinates itself. Mendel controlled this pollination when he was doing things so that he could carefully keep track of what plants were breeding with other plants. So he would use like a paintbrush and take the pollen off of the plants and then pollinate the plants that he wanted to so that he could experiment with different outcomes. To keep track of this, he started to use some terminology, and the terminology that he came up with is terminology that we still use today. He started with the terms parents, first filial generation, and second filial generation. First filial generation is the generation that comes after the parents. The F2 or second filial generation comes from the F1 generation. When he was doing his experiments, what he noticed is that some of the offsprings seemed to produce certain characteristics. And when he noticed that these characteristics always seemed to be reproduced from the same plants, he would call these true breeding plants. Other traits seemed to disappear and reappear in the future. It would be like, if we would think about it today, say 2 brown-eyed parents having a blue-eyed baby, where a trait was not present in one generation and then reappeared generations later. Mendel noticed that with these different traits, the F1 generation only showed one of the traits and some of the others seemed to disappear. When 2 of the F1 plants were crossed, the F2 generation was able to see some of those traits in about 3/4 of the population. We're going to work with some of these numbers. Once we start doing some of these genetic crosses and looking at them, it's going to be a lot easier to see how sometimes crossing certain things with other things result in our outcomes. So what were the factors that Mendel was studying? He was looking at dominant traits and recessive traits. The dominant traits were the traits that seemed to dominate over other traits. The recessive traits may disappear in the F1 generation, but reappear in the F2 generation. Those are what we refer to as recessive traits. Dominant traits dominate, recessive traits, you need 2 genes in order to have that trait show or express. We still use these terms dominant and recessive traits today in genetics. So when we look at the things that Mendel was studying, he was actually studying genes. And the things that he was looking at, like the color of the peas or the shapes of the peas, they were actually determined by one single gene. And a gene is a segment of DNA that codes for specific traits or characteristics. When we talk about this time period, this was very revolutionary because DNA wasn't seen as a thing, and he is really someone who made groundbreaking discoveries, connecting the traits that he did and really beginning the study of DNA. When we talk about genes, we also need to talk about alleles. Alleles are different forms of a gene. So like a gene for brown eyes and a gene for blue eyes are different alleles. The alleles actually separate independently when the gametes are forming during meiosis. So it is random what gets passed on. When we are using letters in genetics, a capital letter will always represent a dominant allele, and a lowercase will always represent those recessive alleles or forms of the gene. Other terms we need to become familiar with are genotypes and phenotypes. So how do genotypes and phenotypes differ? Each organism gets 2 copies of the gene, one from the mother, one from the father, with either dominant or recessive alleles. The genotype is the type of gene that the parents pass on to the offspring, and we write those in the letter format. The phenotype, think pheno, physical. Phenotype is the physical expression of the gene. So it's actually how we would describe something. So it would be tall or short, brown hair, blonde hair, brown eyes, blue eyes. The phenotype is the physical description. So when you look at this below, what you see is you see a capital B, little b, or big B, little b. So one was received from the mother, one was received from the father. This individual would have one dominant allele and one recessive allele. The phenotype is going to be whatever the dominant trait is, because once a dominant trait is present, it overshadows the recessive trait. So, to wrap this all up, Gregor Mendel, father of modern genetics, he used pea plants to learn about heredity. He suggested that some traits were dominant and some were recessive. And even though he was studying the phenotypes or physical expressions, scientists now know that the genotypes were actually present and they were combinations of different alleles. This is just the start of our genetics lesson. We're going to go into more depth as we move on. So let's talk a little bit more about our genotypes and phenotypes and how we can figure some of these things out. The genotype is the letter combination. So it tells us what is the dominant traits, what are the recessive traits, and which one of those are present. They are always represented with just letters. So in our example, it is big P, little p. Our phenotype is actually what the trait actually looks like. So in this instance, our phenotype is actually purple. So purple is the dominant trait. So as soon as we have one dominant trait, that is what is expressed. We can see the phenotype, but not the genotype. In this example, the other genotype that we could have that would give us a purple flower would be big P, big P. Big P, big P, big P, little p, both result in purple flowers. So we can see this here. Big P gives us a purple flower. This trait, when we see this is referred to as homozygous dominant. Both alleles are dominant, so they're both capital letters. Homozygous means that they are the same. Dominant means that they are the dominant trait, the trait that is expressed, so they'll both be capital letters. Big P, little p is considered heterozygous. Hetero means different, homo means same. Think about the root words that we have learned. Heterozygous means that two different alleles are present. So we have a dominant trait and a recessive trait. This still results in the dominant trait being expressed, so our flowers are purple. For the white flower, we have to have two recessive traits present. Again, this is considered homozygous because the alleles are the same, but they are both recessive traits, so it is homozygous recessive, little p, resulting in white flowers. So if purple flowers are dominant, any genotype with the dominant allele will be purple. In order for it to be white, there needs to be two recessive traits present. Let's try a little bit more prediction with genotypes and phenotypes. So if we have round seeds being dominant and wrinkled seeds being recessive, what would the phenotypes be, or what is physically expressed for these following genotypes? So what would it be for big R, big R? If you remember, since round is dominant, we're going to use the capital letter. So big R results in a round seed. Big R, little r also has that dominant trait, so that means big R, little r results in round seeds. Little r are both recessive traits, so it results in the seeds being wrinkled. Now, we can use something called a Punnett square in order to predict the outcome of crosses between two parents. A Punnett square is a quick way, it looks like a checkerboard, for us to see what the possible outcomes are between two parents. So what we do is we write one parent's alleles on the top of the 4 square boxes and one parent's down the side. There is only one letter above each box. And what we then do is we bring the letters back together inside the boxes, bring them down and bring them over, and this gives us the probability of the offspring's appearance. And it gives us all of the different possible combinations of alleles from two parents. We will be practicing and working with this more in class. So let's look at a Punnett square example for the flower colors. So we have two heterozygous purple flowers being crossed. So when we set up our Punnett square, the mothers are across the top, big A, little a. The fathers are down the side, big A, little a. We then bring those letters back together inside of the squares to try to predict what the offspring will look like. We bring the big A down and the big A across in the first square, resulting in big A, big A. We do the same to result in the Punnett square being completed like you see. So what this tells us is that the genotypes of the offspring will be 50% heterozygous, big A, little a. 25 will be homozygous dominant, big A, and 25% will be homozygous recessive, little a, little a. Our phenotypes would be 75% purple and 25% white, because in 3 of those squares, we have big A's. Each of those squares, think about it as 4 quarters equal $1. Each of those squares is worth 25%. So our phenotypes result in 75% being purple and 25% being white. That's the end of our basic genetics lesson. We will move on to advanced genetics next.